I'll talk about B-cell epitope predictions now. We had a really talented postdoc that's done great work on this, and then he got hired, and he's now in India and gone. So now I'm presenting this to you, and I'll try to do a relatively good job, but I won't be able to keep up with what he did. So B-cell epitope predictions. I'll start with a bit of biology recap. You already did some of the 3D in the context of the 3D structure stuff that Morten was showing. You've seen some of this, but it never hurts to repeat. And I'll go through the prediction tools we have in the IEDB, which are roughly split into two bits. One is the linear sequence-based prediction tools, and the other one is discontinuous 3D structure-based prediction tools. And then I'll just very briefly, actually, Morten has already done some of this, the antigen antibody structure modeling. The antibody structure modeling talked about an antibody protein docking. So starting with the biological background, you have seen this several times now. So this is an antibody where you have the variable domains up here, the heavy chain and the light chain. And in this case, this binds to an influenza protein, which contains of two chains, the HA1 chain and the HA2 chain, which form a complex. And then here you have really the binding interface. And this illustration mainly is trying to say is that antibody epitope recognition is complex. And just pretending that you have like a small peptide being recognized by an antibody, which you do to a large extent in the case of T-cell epitope recognition, where you bind to peptides that are small fragments bound to MHC. This is very different when you're looking in the context of an antibody recognizing a part of a protein. So what we call the epitope are the residues on the antigen that are in direct contact with residues of the antibody, and that part of the antibody that forms this contact is called the paratope. So really you're trying to look for a protein-protein interaction where the epitope on the antigen binds the paratope on the antibody. So when you look in more detail how this binding site is formed, you typically have a discontinuous epitope. So you have individual residues on your protein chain that are really making this context. And you might have loops that separate these in the linear sequence of an antigen. These can be quite far apart. And they come together in the 3D fold of the protein to really form this epitope. Now, you have other examples where you say you have in context with the alpha helix, you can say this, when you synthesize this whole region, and you can test that and you get a binding, and we could say, okay, this is a linear epitope. In reality, if you would look for the binding site here, only the residues that are pointing kind of to the outside here would be the ones in context. So while we often say something is a linear epitope, because essentially you can synthesize the linear fragment and get binding, if you would look really into the molecular detail, typically those are still, in reality, discontinuous residues that are in direct contact. So if you're being as strict as that, you would say, well, 90% of all B-cell epitopes are really discontinuous, and that there's gaps in the linear stretch of residues that are being bound. Now, at the same time, you can argue differently if you can call something linear, if you just are capable of synthesizing a linear fragment that is able of binding. So it's a bit of a semantic issue what you consider linear versus what you consider discontinuous. Also, obviously, in much of the data I'm showing here for educational purposes is 3D structures. When you have these crystal structures available, for the vast majority of data that we have in the IEDB, people have not done all the 3D crystal structure complexes of antibodies with proteins. What you typically have is things like ELISAs, where you measure peptide recognition by antibodies and you get a signal yes, no for your whole fragment. So much of the data that we have in the database is about linear peptide segments where we don't know which residues exactly are in contact with the antibody, and then that's why much of the data looks linear in the IEDB, just because those assays are easier to perform, even if we know that in reality the real epitope most likely is a 3D structure and has a discontinuous in nature. So this is the website where you see essentially in this tab for B-cell tools, you have the linear and the structure-based, well, actually, no, so you have as the first one, our linear epitope predictions. And so when should you use them? So here, more than anything, we would really say you should first look, has anybody done the experiment? That is, for B-cell epitopes, even more important than for T-cell epitopes, we think, that if you have experimental data, why predict? The other reason might be if you might also be just interested in other things that are not tested in the IEDB, and you want to have that kind of information. So in terms of sequence-based predictions, the first ones that we have implemented, and I'll show those later, are essentially a re-implementation of historic methods that have been that were the first ones that were used to identify essentially the features associated with antibody recognition. And so there's some things like beta turns, surface accessibility, flexibility, some antigenic propensities, hydrophilicity. So these were all essentially scales where people that were previously defined, and hydrophilicity is a good example, where people had for every one of the 20 natural residues, people have calculated or maybe even measured a hydrophilicity property of that residue, and then you can look at stretches of amino acids in a protein that are antibody epitopes, and you find that they are typically more hydrophilic than others. Now, what are you predicting with that? Many of these are actually very highly associated with regions of a protein that are on the surface. So you're really having, to some extent, a poor man's version of a prediction which residues are surface accessible on the protein, because those are obviously the ones that are more likely to be bound by antibodies. And again, like, we have implemented these to a large extent because they were reported in the literature, but there was no interface in which they could easily be run. So that's why they are there. And you essentially, and I'll show this in a second, you provide a protein sequence, they scan through with these properties and identify regions in your sequence where you have a cluster of, say, hydrophilic residues. Now, one of the important things if you're doing that is to limit yourself to the protein regions that are actually even accessible for antibody binding. So, for example, many of the proteins have, say, signal sequences at the start that are going to be cleaved off before a protein comes, say on a, or you have a membrane standing part of a protein and there's an outside loop. You should obviously focus on the things that are actually accessible for antibody binding. And then within those, these scales tend to work. So the other set of linear epitope prediction tools are based on machine learning rather than having a preset property of amino acids that you're correlating with epitopiness. There's a number of machine learning algorithms that have been developed to try to learn based on the sequences that we have identified to be epitopes, what is essentially the underlying motifs or so in there. And the method that we are primarily using is BepiPred, and there's two versions of that, 1 and 2. There have been a number of other measures out there, and I later have some benchmarking data sets. We have not found any improvements over the BepiPred performance by these other methods, so they are not implemented on the IEDB, and they were not easily available for us to implement. The prediction accuracy of these methods is very limited, and I come back to that in a second again. So starting here with this prediction tool, this is how it looks like. This looks very familiar, what you have been seeing all day in terms of T-cell epitope prediction method. There's a sequence input. Here we also have the ability to just go straight forward for a UniProt or SwissProt ID here, and then it retrieves the sequence externally. Then here you have a number of these different scales that you can select after you have input your protein sequence. The default is BepiPred. It should actually be BepiPred 2.0. I don't know why we do as a default an out-of-date method. So I would actually recommend to choose the 2.0. And in each of these, if you click here, you go to the Help tab, where there's a description of the underlying paper from which this method was derived, and the description in the case of the scales directly what it is that is being used. And typically all the methods differ a bit in terms of what is the recommended average window of residues that you're looking for when you apply these methods. So the reference tab here, you're going through the individual tabs here now, contains specifically each of the papers that this is based on. So as an example, we're putting in sperm whale myoglobin, and this is the sequence, which comes here from the SwissProt record. And using BepiPred and submitting, you're getting here now as a function of the length of the position in the protein sequence. You have this aggregate score, which essentially goes over a window, sliding window of amino acid residues, and calculates this BepiPred score. And then what BepiPred does, so there's a threshold here, which is, in this case, is set to 0.35, and you see there's the yellow regions that go above that threshold are indicated here as the regions that are of potentially immunogenic. And that calculation is then used to identify, I mean, here you have essentially just a repeat of these values in tabular format. What BepiPred does as well is kind of summarize then the consecutive residues of what are considered to be linear stretches that are potentially immunogenic for antibodies. Here is now an example of the kind of scales you're getting with 3 different of these scales, the Chu and Fastman predictions, the Kaplus and Scholz predictions, and the BepiPred predictions. And what you see in this case is actually that you have similar regions in all 3 cases being flagged as likely to be epitope enriched. And when you actually, one of the recommendation is that if you're looking at these things in conjunction and asking for regions that all your different methods are identifying as potential epitopes, you might have a higher confidence that these are actually real rather than relying on single methods. Now, when we did a large-scale comparison of these different prediction tools, the performance that we got, this is still with BepiPred 1.0, was 0.6 in terms of AUC. As Sinu showed you, for T-cell epitopes, we are at 0.975, so where you're really close to 1 of perfect. With 0.6, you're suspiciously close to 0.5, 0.5 being random. So these methods are nowhere as good as you can get with T-cell epitope predictions. They are still helpful, but this is not something that you would want to bet your house on. They essentially allow you to prioritize and to interpret, essentially, when you're looking at a protein. But yeah. So, and one of the reasons, as I showed you before, is like these methods treat the whole protein sequence just as this linear string. And antibodies do not recognize a linear string of amino acids. They recognize regions in the proteins in a 3D fold. So the whole performance of these methods and the sincerity of these predictions is much improved if you're starting to look at your protein in 3D space. So ideally, you want to have your antigens, so where you're looking for antibody binding sites, to be identified and characterized in terms of its 3D structure based on X-ray crystallography or NMR or EM, whatever data is out there. Now you have in the IEDB itself, for things where you have an antibody bound to a protein, you can directly look at those records, and you can, as Morten showed you, use the Scepter tool to use them. This is primarily for benchmarking if you're looking for antibody-antigen complexes. Now, if you want to have a new protein where there aren't any antibodies bound, you should try to get the 3D coordinates directly from the PDB. And I think I have something about that later, yeah. Now, once you have that, you can use the 3D structure of your protein sequences to use other methods, specifically DiscoTope and ELIPRO, which we're going to have implemented on the IEDB. There's a number of other algorithms out there that unfortunately were not publicly available for us to implement, but they exist, and there's publications for them out there that you might want to consider as well. And then there's general purpose protein docking algorithms that could in principle be used, because that's essentially what you're looking for in terms of a problem if you're that you're trying to dock antibodies against your protein. And yeah, So specifically talking about DiscoTope, that is an algorithm that was trained originally on 75 crystal structures of antibody-protein complexes. DiscoTope 2, which came out, so DiscoTope was 2006, DiscoTope 2 was 2012, took into account that there isn't just one epitope, there's often multiple epitopes in an antigen, and also took into account, I believe, the homodimers and heterodimers, so that parts of the proteins are actually not even accessible for antibody binding in the training. So what these methods do, they start off, what DiscoTope does, is it assigns each residue a score based on a linear combination of multiple values, such as the hydrophilicity of the values. This is one of the linear scales, the amino acid occurrence in antibody antibody epitopes compared to overall residues on the surface. And then here, this is where you need the 3D structure. It looks for essentially that these residues that you're marking in your 3D complex, they must be coming together. You're looking essentially for a patch of residues on the protein that overall, and not in the linear sequence, but in the 3D structure that comes together and looks like an epitope. So there was an improvement going from 0.71 to 0.73 with DiscoTope 2, and this is a useful prediction to make. So these were the papers that go with it. This is how you use DiscoTope. So you start with having to enter a 3D structure of the antigen you're looking for, and then in the PDB, as you might know, so the PDB always has a 4-letter code for a 3D complex. And in that complex, you might have multiple protein chains. So you need to identify which of the specific chains do you want to make a prediction for. And then, so this is actually how in the PDB you can search for names or sequences or to get these identifiers. Okay, this is just jumping through it. In the searches, you can search by sequence and then pick up structures where that protein has to be crystallized. You can also search by name or any kind of other thing that you care about. A limitation here is clearly that if there is no crystal structure of the protein that you care about or something highly homologous, then this tool will not be usable. So this is, again, on this PDB side, you can grab out the sequence for the structure that you found and the whole information in PDB format, which is containing the 3D structure. Okay, then here, also you have the breakdown of identifying, in this case, you have 2 chains, A and E, that are belonging to this Plasmodium falciparum protein. Okay, so if you don't have, I don't learn apparently. So if you don't have 3D structure available, the most reliable way of getting a 3D structure of a protein that there isn't one is looking for one that is has a very high sequence identity, and then do homology modeling. That is not something we have implemented on the IEDB website, but there's a number of web servers that do it, and there's kind of an overview page in the IEDB you can go through recommending different servers that you can use for this purpose. We actually originally okay, so once you have this data, you can go back to DiscoTope, then here you enter your the PDB record, or you could actually, if you had a file based on a homology modeling tool, you can upload that directly. You enter your chain. This is the version of DiscoTope to be used. 2.0 would be recommended, and you hit submit, and then you have here an overview of from these papers different thresholds, how different thresholds relate to different levels of sensitivity and specificity. As you see, the thresholds here have changed quite a bit from DiscoTope 1.1 to 2.0. For 2.0, the threshold of 3.7 is associated with like 47% sensitivity and 75% specificity, which is we would normally recommend as a default threshold. So you submit this, and then you're getting here this these values, and you can see essentially this is the 3.7 threshold, and everything in green is a positive prediction. This is visualized here on the linear sequence, but you can go through different views. So you can adjust your threshold if you want to have other estimated values for sensitivity and specificity. You can save your prediction as a CSV file. You can do a table view of essentially this data, which is just like for each residue then listed the specific score. And then more interestingly here, these are the three that are better than your threshold, and you can look at them on the 3D structure. So you essentially then the parts of the protein that are better than your thresholds are highlighted. Does this work? Oh yeah, it does. And Stopmill did this, and I'm sure he had something in mind that I should be saying right now as this thing is moving. Here. So yes, you can click these things, and it highlights in your 3D structure the individual residues from your prediction, so you can look this way directly at what is it that is above your threshold or not. So this is the DiscoTope method, which essentially, again, like it looks for, it combines in analysis of what kind of residues are associated with epitopes in general, then looks for surface patches that are associated with it. The ELIPRO method is an implementation that actually Janet Thornton originally suggested, which is kind of like a geometric interpretation of what properties are associated with epitope antibody binding sites in proteins. Namely, you can approximate, essentially, you can always approximate a protein as an ellipsoid. And what she found is that when you do that, kind of the parts of a protein that are most protruding from that ellipsoid estimate of the protein shape are highly likely, or much more likely to be epitopes than parts that are kind of hidden. So what is ever the most out protruding from a protein is more likely to bind antibodies, and that actually makes intuitive sense in most cases. So this was a method that was originally published already in '86, and essentially we just took that algorithm and implemented this because, I mean, in '86 there weren't that many web servers around, so can't really blame her that it wasn't implemented back then. And the kind of interface is very similar to what how DiscoTope works as well. You identify the structure based on the PDB ID. Then you have here scores. As always, unless you have a clear reason, I would stick to the default values that are already set. Then this is the, again, like this is, okay, sorry, there's a score threshold for the minimal protrusion, and then the maximum distance is how much, in terms of grouping these different residues, what is the maximum distance that you want to consider. So once you run this with these values and click submit, you're getting here in this case, so as you saw, you didn't enter a specific chain here, so it actually runs the algorithm separately on chain A and chain B, and you can look at the one of interest, and then here get some similar data back to what you saw for DiscoTope, where you have the regions in the chain essentially that are grouped together as linear stretches within this protein that are potential antibody binding sites. And then these are the discontinuous epitopes that really only come together in the 3D fold. You can, as before, use these view features to go jump to the 3D structure and visualize then the parts of the protein that are predicted to be potential epitopes. And as before also, oops, you can look at the same data here in a table view. So summarizing this, linear and discontinuous conformational epitopes can be overlapping and depending on method of discovery. Okay, so what does this mean? Again, like if I started off by saying, so we are sometimes pretending that there's a clear distinction between linear and discontinuous epitopes, but in reality, most of the things that we consider a linear epitope. If you have the 3D structural information and know exactly where the antibody binds, the vast majority of epitopes is discontinuous. Conformational is again like we are considering that the epitope only exists in the 3D fold of the protein, which again is the case for the majority of epitopes. And the reason why we have so much more linear epitope data in the IEDB is largely driven by that those are experiments that are much easier to perform than 3D structure crystallizations of antibody antigen complexes. The traditional B-cell epitope prediction methods, such as hydrophilicity scales, really are to a large extent predicting surface accessibility of regions in the protein. And if you have the 3D structure of the antigen, that is definitely the preferred way to go to predict epitopes, and predictions can be further improved. So this is where I had a break in my slides, and this I would like to use we are doing very well on time. Okay, so then why don't we do a 5-minute break and start again at, is it 3 or Morten?
Ask one question, do you want to have questions in the end?
No, go ahead.
So for you said in the IEDB that you have multiple chains, you run them individually. Do you mean that?
Yes. Am I thinking that that's a good idea, thinking about it? Probably not. You would want to ideally, so specifically if you have a protein complex of 2 chains binding together, the part that is where the 2 come together is most likely not accessible for binding. But the way it's currently done, yes, and I think this is the ellipsoid modeling relies on having one continuous chain to then estimate the ellipsoid. You could imagine that why not do this for the whole complex? Yeah. And this is Janet's original method. So this is also one thing where we have never really tried to optimize it. We implemented like she had it.
Because DiscoTope 2.0 did exactly that. The DiscoTope 1.0 did each chain by its own. 2.0 did it as a complex to avoid that you have an unavailable surface being predicted as being an epitope.
There was more questions, yes?
Can you hear me?
Yeah.
Okay. So you talked about the scoring system in DiscoTope. It takes into account sensitivity and specificity. So isn't there some compromise on sensitivity with the threshold value of -3.7?
So as you can see here, with the threshold, the first ones, so here you have low sensitivity and very high specificity. And as you're increasing the stringency of your, so this is, it's the higher the better, right? So here you're trying to pick up half, this is based on on the performance data, right? On the cross-validation or blind set evaluation. So here, to get at this threshold level, you're still retrieving half of your epitopes, and your predictions have 75% specificity. If you are more stringent, you tend to lose more and more of your epitopes, but your specificity goes up. So whatever is still above this threshold, you tend to get very high likelihood of them being real, but you're also missing a lot. So yes, there's definitely a compromise.
So, in our studies, we use the mouse models in our research. So one of the critics we often receive is, what do you do with this information, right? Whatever we gain from the animal models. So suppose if you identify a B-cell epitope or a T-cell epitope, and if you want to provide some translational significance, based on that information, and if you go and dig through the literature, maybe by using your database, asking the question whether it is relevant to humans. Have you done some studies to show what we learn from the animal models may be applicable to humans?
Yeah, so I'm sure there's other studies. I'm very familiar with one of the studies we did, which was about antibody responses induced by smallpox vaccination, where the first studies were done were all in mice. And we identified epitopes there, then from essentially made murine monoclonal antibodies from vaccinia virus, which is the smallpox vaccine, immunized mice, and did characterize something like 20 or so different antibodies. And then it took us a while, because making mouse monoclonals was easy, but human monoclonals was hard. And so 3 years later, we had a bunch of human monoclonals come in, and lo and behold, they bound the exact same sites. So actually, I would think that the mouse model for antibody epitopes, as long as the viral infection that you're studying is kind of similar, which often is not the case, but ultimately, because it's a structural constraint to a large extent of what is really available for the antibody to bind, and that is not so different from mouse to humans. I would be much more skeptical about T-cell epitope cross-reactive. Definitely, if you have a mouse with a mouse MHC, that specific peptide that you're finding is not going to be as relevant. So for antibodies, I think there's a pretty good overlap of marine antibody binding sites and human antibody binding sites. Okay, so why don't we do the break until 3?
I think that you only have 10 slides left, so you should just push through.
I should just push through.
Yeah.
That's good.
10 slides. You
Okay,
got it.
yes.
All right. 39 is where you are.
I don't know what I'm doing, I'm not sure. Okay, so these are examples that we had prepared how to use the tools, and I believe what Swapna did last time, what I'm definitely trying to do, is going to go through them, because I do remember when we did them the last time, it greatly showed off how it can be difficult to use the PDB, and that's not what we are trying to show here. We're trying how to use our tools. So the goal would be to identify essentially antibody epitopes in dengue 2 virus enveloped glycoprotein. So, and we wanted to use the BepiPred and the DiscoTope tools to do that. So, and this is obviously, it's a really relevant dengue virus, as you guys know. I mean, there's 4 different serotypes, 1, 2, 3, 4, and antibodies are known to be contributing to clearance of and protection from subsequent infections of the same strain. And there is definitely a lot of vaccine development going on. One of the goals here would obviously be to try to see which parts of the virus are potentially accessible for antibody binding and should be included in such a vaccine. So if you go to the PDB, this has been crystallized, and you can then download the crystal structure and sequence of dengue 2 virus glycoprotein from the PDB. So, and this is the code. If you want to, please follow along and do this. So if you do that, then and you get the sequence from the PDB and put that into the tool. This is kind of what you're getting with the default thresholds. And as you see here, with the default thresholds, you're essentially identifying half or more of your whole protein residues in your protein to be above that default threshold, which is not a very useful prediction. You might as well make the whole protein sequences now. So by upping the threshold to here 0.9, which corresponds to 90% specificity, you still have a number of regions here that fall above this threshold, so these could be considered to be good candidates. If you enter the 3D structure into DiscoTope, this is the default output that you're getting with the minus 3.7 threshold, which was our default threshold. And you see here, there's a few regions that make it above that threshold. And so these would be then, according to DiscoTope, the ones that are more likely to be epitopes. And as we've seen before, DiscoTope outperforms BepiPred. So in a case like this where you have both available, normally we wouldn't even look at the BepiPred predictions, but you don't always have crystal structures of your antigens available. So it's that's why there's still a reason for having these linear epitope predictions. So when you look at this here, actually in DiscoTope, you notice that your sequence stops at before 400, and here your sequence goes up until 420. And the reason for this, 422 compared to 391. I have the numbers here. So the reasons for this discrepancy is actually something important to keep in mind whenever you work with crystal structures. In the PDB, you have two sets of sequences. One is your sequence record. That is what the person who made the crystal structure expressed and put into their crystallizing reaction, and you see the whole protein. And then you have atom-seq. That is essentially the reconstruction in the crystal of the amino acid residues that you could see. Because when you crystallize a protein, if you have flopping ends of your protein, and you typically have that, they will not always, in order for something to be crystallizable, it has to have the same conformation. All the proteins you have in your crystal, floppy ends are going to be gone. So you have things here that are missing. This can be, it's not the case here. I don't know if Morten touched on it, but this is one of the fundamental problems often with a crystal structure, specifically if you have, this might be a CDR region of an antibody. If it's not in a tight conformation that might be flopping around, you're missing those residues. Whenever you have something that is not in a stable structure, you're going to be missing them. So when you look at the results, and we are looking here now in the BepiPred and the DiscoTope predictions, you see you have a number of, so the real epitope, obviously we chose one where we know the answer, when we can get that from the IEDB, are the correctly, these green parts of the protein where you really have the antibody binding site. And you can actually see here that this is nicely overlapping with the region that both DiscoTope and BepiPred picked. But as I also showed you, I mean, DiscoTope has much fewer predictions, and then they tend to overlap here with the correct binding site, so that is encouraging. There are additional residues from the IEDB that are, This is using essentially the immunome browser and identifying where in dengue virus did you have linear epitopes that kind of stack up. And if we use that as a prediction method for the 3D structure, we also find overlaps here in terms of the regions where linear epitopes were mapped and regions where the specific antibody bound. So that's essentially actually the comparison of the methods that we have. And then there's the, what we are hoping is going to be improvements to the antibody epitope prediction field in general. So this was, I think, one of the first methods that went away from just asking the questions, if I have a protein, where is my antibody binding site, to saying, okay, if I have a protein, very often there's going to be multiple antibody binding sites. And those multiple antibody they are going to be determined based on the specific sequence of the antibody. So really asking for, given a protein, where is the antibody binding site is kind of not a very well-posed question, because different antibodies will bind in different sites. So in the case of T-cell epitopes, we are typically asking, where is the epitope, a T-cell epitope that is restricted by HLA-A0201? That is already drastically limiting what we're looking for. We're not just saying what could be a T-cell epitope in this protein sequence, because if you can imagine all kinds of different MHC molecules, all with different binding specificity, and different things would be positive. If you're, with the antibody, it's kind of the same thing. We're trying to ask for a more precise question. So if you do have a monoclonal antibody, can you predict where that monoclonal antibody will bind? That becomes a much more interesting question. And this PEACE algorithm that was actually coming from the vaccinia data that I mentioned earlier, where we had a whole number of monoclonal antibodies that were binding different parts of the D8 antigen vaccinia virus, and then asked essentially, can we distinguish where the different antibodies bind? And so that worked in this context and is something that we believe should be getting better and better as more data becomes available of people doing high throughput sequencing of antibodies with known specificity, where we are hoping to essentially learn these rules, how specific antibodies bind to specific sites in a protein. To my surprise, we haven't gotten a large amount of epitope-specific antibody sequences. And one of the reasons is that actually much of the data that comes out right now in the literature is not epitope-specific, but antigen-specific. So people are using an antigen to pull out B cells that have a certain specificity, but they don't go further and saying this all binds this particular region in a protein. Because actually for most of their applications, they are fine by just saying this is in whatever, binding a certain protein in HIV, say, and not everybody cares as much about epitopes as we like. So this server exists, and these are, to a large extent, I think, still proof of concept predictions. One of the issues is, I know Morten has also been working on similar tools like this. Some of these are harder to implement as servers because there's a lot of 3D structural manipulation necessary, which are more computationally intensive than things like MHC binding predictions. So there's been a number of benchmarks coming out. So this is probably the oldest one they have, 2008, 2012 is better. Here you saw like DiscoTope 2 performed better than DiscoTope 1, and yeah, so a number of other tools are kind of in this range. That was kind of where we were at here. And so why is it so low again? I mean, the reasons here are other ones, like very often we are asking for a whole protein for where's the antibody binding site, and then many parts of the protein aren't actually accessible for antibody binding. Then we are still lacking, definitely because the crystal structure generation is more labor intensive and more rare than say, defining a peptide T-cell epitope. And then the other reason here that ultimately this kind of question, where you're asked to determine in general if a region in a protein can be an antibody epitope, is not as well posed as saying, is this a T-cell epitope in the context of a given MHC? That is the last slide I have, and Sheridan was right. That was definitely faster. Yes, please. So that's a very, very, very good question. It really depends a lot. So we've had long debates with different people. So for example, many of the viral people will be referring to, will have as their gold standard the capability of a virus to neutralize a virus. And then you'd look for an escape mutation, ultimately saying, when I mutate this residue, the antibody loses neutralizing ability, and then you do this systematically, and then you can map, these are the residues that are critical for neutralization, and the viral people will say that is the only relevant data, because it's. Then you have people who are saying, well, I'm using epitopes as a diagnostic tool. I want to put them on an ELISA plate. I want to measure it tight, and I want to know, does this person have hepatitis B or whatever you're trying to diagnose, using a serum diagnostic? And they're saying, this is my criterion. And these are going to be completely different. But it's a perfectly valid reason to want to know what these peptides are that work good as a serum diagnostic, but they are not going to be neutralizing. And then the 3D structure people who want to know how does antibody, antigen, they say I want to know the 3D structure of the whole complex, otherwise I don't know anything. So unfortunately it changes. The next thing about what we have found, in we had, when I had run this vaccinia virus antibody epitope mapping study, there was multiple other contracts that were running in parallel. And the thing that actually worked the best, and every one of the contracts ultimately did it, was mass spectrometry fingerprinting, which is with using deuterium exchange, which is a fascinating thing. Where you have the protein that you have your antibody. You know that the antibody binds the protein. Then you you soak it in deuterium, with or without the antibody present, and then you essentially cleave it up. Do mass spec, and you identify which parts of your protein have taken up deuterium, which ones didn't. And the ones where the antibody bind, they will be protected, and they won't have it. The great thing about that is, in contrast to 3D structures, that always worked. So if I were to do this study again, that's what I would have been doing. I think that was the best way to get both linear and discontinuous epitopes in a very reliable fashion, yes.
Though of course the cryo-EM people are
Yes.
are now doing
Yes.
really a bang-up job of actually visualizing the antibody bound to the antigen, which is almost unbelievable to me.
Completely right. And I mean, not that many people currently have the access to these instruments, and it works best if you're studying the same protein kind of, but yeah, it's fascinating. And the resolution there is going up in leaps and bounds. It's fascinating. It used to be the cryo-EM, you could barely just see something, and now you're really down to residue, amino acid residue resolution for.
One follow-up question. I see in a big picture as to the basal prediction, but I'm wondering why the length of the epitope should not be emphasized in the basal prediction tools.
Length of the
Yes. You know, like in T-cell epitopes, we say, 15, 20 amino acids. Suppose
Yeah.
if you were to choose some bigger fragment, would you get the same data or output? Well, given that,
So this-
continuous, discontinuous.
So I guess the reason is when you, and Morten should probably be weighing in, but when you look at some antibody binding sites are gigantic, and there might be 40 residues in there that are in contact. If you have like kind of a flat surface, then you have another antibody that barely hangs on to the tip, and you have a single residue or two. It looks like, so it is, I think, broadly more variable that for T-cell epitopes. Morten, do you have a comment on that?
No, I can only say I agree. And this is actually when we are trying to develop these tools from the antibody sequence predicting the target, the most important feature we get is the size of the paratope. The paratope is the antibody interface, right? That should match the epitope site on the antigen. And if we, from the antibody sequence, can predict the structure of the antibody and getting out just the size of the paratope, that's the most important predictive parameter we have to locate the epitope on the antigen, because that you have to kind of find something on the antigen side that has the same size as the paratope with the properties of being exposed and having protrusion. This allows us to at least do some predictions, not very good, but better than random, just from the size of this kind of complementarity between the paratope and the epitope.
And I guess like in that, the biological underlying reason is obviously for T-cell epitopes, you have the MHC that serves as the normalizer. You have to have a certain size to even fit in the MHC molecule. And then the TCR engages that complex of MHC and peptide. Versus antibodies, I mean, they evolve and mature to hang on to one binding site, and they can look vastly differently versus all TCRs pretty much look the same.
In a big picture, what is the length of the paratope based on your experience?
But this is a patch, right? It's a 2D, semi-3D thing, right? And as Björn said, they vary a lot. I mean, there is maybe, on average, you have 25 residues involved in the paratope, something like that, 30 residues involved.
Any more questions? Morten?
Just one comment to I think so the field of predicting stuff is moving a lot these last years with all the deep neural network methods coming out, both in the T cell and the B cell field. And then just I think a word of caution, because the IEDB tends to incorporate as much as we can of good tools, and you might not find many of those new tools in the IEDB because they have performance values like of a B-cell epitope predictions. There are papers being published with an AOC of 0.95, right? And you saw Björn's benchmark in the performance values are 0.75, right? And I don't, hopefully I'm wrong. Someone will invent the magic tool to do it right. But I don't believe in quantum drops. Performance will improve steadily from 0.75, 0.8, and then hopefully in some years we will get there. But I don't believe in these magic jumps overnight from bad to be perfect. When you read these papers, I mean, and you wanted to kind of try out tools, I think you should just have a word of caution. Don't believe in performance values bigger than 0.9 for predicting B-cell epitopes yet. It's not correct.
So, I'm quite interested about the discontinue the epitopes. So, have you ever think about this? Because I'm the virus guy, so most time I focus on like a VLP virus like particle. The particles, as you know, maybe assembled from like 60 to 100 something different or same subunits. Have you ever considered some kind of discontinue the residues from different subunits, even the subunits exactly same, but just different orientation on the VLP or the capsid?
Yeah, so I mean, definitely in the database we have them and we find them. This is then going also towards the mixture of cryo-EM and 3D structures. Typically, if you have a 60 subunit viral, you will not be able to crystallize that. You might be able to reconstruct that and the symmetry of the different subunits within it from cryo-EM data and then merge it together. I do not believe, for something like a homodimer or something like that, the predictions will work. For something like a 60 subunit complex with different symmetries, it'll not. So it's definitely an interesting problem. Will?
Just a comment, even for dengue, for example, the envelope does form a homodimer. The most important neutralizing epitopes are ones between adjacent dimers, because they inhibit the reorganization of the envelope for infection. So it would be really nice to be able to stack those dimers in the correct orientation, so you could predict.
And you have all the other issues with the breathing viral.
Yeah, of course.
It's ultimately, you really want to have the
dynamic structure, but
but
if you only have one at a time, but the ones the whole undersurface and most of the sides aren't going to be relevant most of the time.
People said about stems, epitopes, but
How do you take into account in these predictions the maturation of affinity?
We don't. So essentially what we are, ultimately all of our models are trained based on antibodies that were isolated that presumably have undergone affinity maturation. So but there is no, that would be really cool. I mean, if you start off by saying this was the germline sequence it's coming from and it looks like these residues have specifically mutated, and there have been experimental studies like this and showing that those mutations are more likely to be in contact because they must be or important for the, so combining the knowledge of which one of the mutation in a given antibody are really mutated, and specifically if you then have like a whole set of antibodies and you see what they share or so, and could you incorporate that? Please invent a tool like that and then we can host it.